Evolutionary Science in the Catholic Church by Reverend Mr. Kevin W. Vogel. Read by Father Kevin W. Vogel. Evolutionary Science in the Catholic Church, Chapter 2, The Science of Evolution. In order to understand the interaction between biological evolution and Catholic theology, one must first understand what evolution is. Most broadly, evolution, like other scientific theories, attempts to interpret objective physical data within a framework that allows connections to be made between individual sets of evidence to create an explanation of the whole. A philosophical consideration of what evolution is as a science will be covered in Chapter 4. The goal of this chapter is to first present some of the main facts that are used in evolutionary theory, and secondly, to describe the different frameworks, theories, that have been formulated in an attempt to explain these facts. The intention is not to give a comprehensive overview of such facts and theories, but rather to give an outline of some of the terms and ideas within the scientific discipline of evolution. Footnote 30. This is an important beginning so as to prevent confusion about scientific facts and terminology. Initial observational evidence. The first major piece of evidence is the observation that there is a diversity of individual life forms. Not all life forms are the same. Not only is there great diversity, there are also similarities between individual forms of life. Based on these similarities, this diversity is characterized into different groups that share similar characteristics. Depending on how general or specific one is with the characteristics, different levels of categorization are formed. The ranks of classification from general to specific are domain, kingdom, phylum, class, order, family, genus, and species. Footnote 31. Even at the level of the closest similarity, the species, diversity is still present in the form of variations in the characteristics of individual members. Footnote 32. The second piece of evidence is that this diversity and similarity in life forms not only occurs within life forms currently living, but also throughout the history of life forms. Small-scale changes have been directly observed within populations of organisms. Footnote 33. For example, two isolated populations of the same species have been observed to develop separate characteristics over time. There is also evidence of long-term changes, especially apparent in fossils. A fossil is any trace of once living organisms, thus giving a record of what types of organisms lived in the past. Many fossils are of species that have no living members, witnessing to the fact of the extinction of some species. At the same time, there are organisms living today that are not found until a certain age in the fossil record. The general pattern of similarities between fossil forms and living forms from the same location became known as the law of succession. As earlier forms of life die off, new forms appear in the fossil record. Footnote 34. This diversity, similarity, and observed changes over short and long periods of time led to the formulation of scientific theories that attempted to explain and connect these pieces of evidence. It was hypothesized that, like the small-scale changes, the wider diversity of life forms was due to the development of previous organisms into new ones, explaining the similarities within diverse organisms with a common ancestor. In the early 1800s, French biologist Jean-Baptiste Lamarck advanced the theory that all species are derived from other species, moving from simple to more complex. Footnote 35. While the short-term changes within a population termed microevolution are directly observable, long-term changes between species termed macroevolution are not directly observable due to the intervals of time between generations. Footnote 36. The long periods of time required for changes from one species to another limit the ability of researchers to directly see the change, so they rely on indirect means such as fossils. The fossil record contains transitional forms of organisms that exhibit traits of both their ancestors and their descendant species. These transitional forms are used to create evolutionary trees, or phylogenies, mapping the relationship between organisms living today and their common ancestors. Footnote 37. It will be shown that microevolution and macroevolution are connected. since microevolution is a necessary condition for macroevolution, but not a sufficient condition. It is important to keep them distinct, since the observation of microevolution does not necessarily imply the presence of macroevolution. Darwin's natural selection theory. Once it is hypothesized that life forms develop over time into other life forms, macroevolution, it must be explained how this could occur. Building on the work of those before him, Charles Darwin formulated his theory of natural selection in his work The Origin of Species, published in 1859. Footnote 38. Darwin began first with 3 observable facts. 1. All species have the potential to increase in population size if all individuals that are born survive to reproduce themselves. 2. What is observed is that apart from normal minor fluctuations and occasional major fluctuations, populations remain relatively stable. 3. The natural resources available to a population are limited and remain constant in a stable environment. Based on these 3 facts, Darwin inferred that since more individuals are produced than can be supported by the available resources, and that the population remains at a stable size, individuals in the population must compete against each other to utilize the resources, resulting in only a part of the offspring surviving to reproduce the next generation. Darwin then noted 2 more facts. 4. There is variation in the characteristics of the individuals of a population. This is the diversity spoken of above. 5. Most of the various characteristics of individual organisms can be inherited by their offspring. From his first inference in these 2 facts, Darwin made 2 further inferences. First, he inferred that the competition among individuals to survive is due in part to their different inherited characteristics. Certain characteristics are better suited for survival than others. The individuals with these characteristics, by the fact that they survive to reproduce, are naturally selected to pass on their characteristics to the next generation of offspring. This inference he called natural selection. Second, he inferred that over many generations of inheriting variations of characteristics that were better suited for survival, the changes would build up to produce new species. This change in the descendants of an organism is called descent with modification. Footnote 39. This initial form of evolution, descent with modification due to natural selection, is termed Darwinism, Darwinian evolution, or natural selection theory. It is important to note that Darwinism, while it is ultimately a theory of macroevolution, applies first at the level of microevolution, which is its necessary condition. Natural selection and descent with modification first apply to changes within a population. Secondarily, they apply to changes between species. The concept of descent with modification is what marks the difference between the 2 levels. On the first level, the populations are modified, while on the second level, modification produces new species. These 2 levels of change remained in the background of the progression of evolutionary theories that will be discussed in the next sections. Footnote 40. Natural selection and variation. The term natural selection actually represents an analogy that Darwin made between the selective breeding done with plants and animals, for example orchids or horses, by humans, and the selection that occurs among organisms that happen to live to reproduce or not. Darwin noted that in selective breeding, it is man that chooses or selects which individual organisms have the desired characteristics he wishes to have in the offspring. While the breeder has no control over the variations that happen to arise, he does control which individuals he allows to breed. Darwin then asked, quote, "Can the principle of selection, which we have seen is so important in the hands of man, apply to nature?" End quote. Footnote 41. While the selection done by man is a personal choosing, the selection done by nature is not a conscious choice, but rather an automatic result of natural events. Quote, "Natural selection is a direct, inevitable, and automatic consequence of basic demographic processes." End quote. Footnote 42. Darwin described natural selection in this way, quote, I have called natural selection, or the survival of the fittest, this preservation of favorable individual differences and variations, and the destruction of those which are injurious variations neither useful nor injurious, would not be affected by natural selection, and would be left either a fluctuating element, or would ultimately become fixed. Footnote 43. One can see that Darwin recognized three kinds of variations: advantageous, deleterious, and neutral. Footnote 44. Advantageous variations increase the chances of survival, deleterious variations decrease the chances for survival, and neutral variations have no direct effect whether or not an organism survives. These variations, according to Darwin, are, quote, governed by many unknown causes. The final result is thus rendered infinitely complex. End quote. Footnote 45. Variations are caused by multiple unknown factors, so that one cannot predict what variations will arise. This is the origin of the discussion of chance and evolution. Variations are produced by chance, which does not mean they have no cause, but rather that the causes are complex. Quote, often chance is introduced as the scientific way of treating situations in which the causal system is too complicated to be analyzed in detail, when there are too many causes that interact in producing the phenomena being studied. End quote. Footnote 46. Neo-Darwinian theory. In 1866, independent of the work of Darwin and others in evolution theory, Gregor Mendel made his discoveries that would later be the basis of genetics. Mendel deduced that the correct mechanism for biological heredity was a recombination of units of inheritance contributed by both parents. Footnote 47. After Mendel's work was rediscovered in 1900, it was around 1940 when classical genetics and evolutionary biology were joined in a new synthesis, sometimes called the synthetic theory or neo-Darwinism. Footnote 48. There are two main elements of this new synthesis. First, the variations, also called mutations, and the recombination of the hereditary units of the parents in their offspring are undirected. This means that a variation or recombination will not occur more frequently if it is advantageous, or less frequently if it is deleterious. The neutral category Darwin acknowledged is not retained in neo-Darwinism. The second element is that a variation or recombination can be determined to be advantageous only after it is selected, only after the organism survives to pass on its variations to its offspring. Footnote 49. Thus, neo-Darwinism, also called the selectionist theory, places great importance on advantageous variations, since these are by definition the ones that are selected by natural selection. Footnote 50. In this way, natural selection is seen as the main mechanism for evolution. Molecular Darwinism. Following James Watson and Francis Crick's work on the DNA molecule in 1953, the field of molecular genetics was developed. Structures and functions of organic molecules began to be better understood. The synthesis of molecular genetics and neo-Darwinism led to molecular Darwinism. Footnote 51. Some of the most important molecular structures for the passing on of genetic material include nucleic acids and proteins. The molecule deoxyribonucleic acid, DNA, was found to contain the, quote, instructions for making and maintaining an organism, end quote. Footnote 52. The DNA molecule is made up of structures called nucleotides containing 4 types of nucleic acids, labeled A, T, G, C. The information contained in DNA is transferred first to RNA molecules and then to produce proteins, from which the different biological structures of the organism are then produced. The order of the nucleic acids in the DNA molecule determines which proteins will be produced. Sequences of 3 nucleic acids contain the code to produce one of the 20 different amino acids that first build up proteins and then the rest of the organism. Footnote 53. The observed characteristics of an organism have their source in the coding sequence of the organism's DNA. The hereditary units described by Mendel were found to be a series of DNA nucleotides, now called genes. The changes found in the hereditary units are changes or mutations in the sequence of DNA nucleotides. There are three strategies by which mutations occur in the DNA nucleotides of an organism: local changes in the DNA sequence, DNA rearrangement, or the acquisition of new genetic material. These three strategies are produced by a combination of four mechanisms. 1. Changes occur during the process of DNA replication. These changes are due to the substitution of one nucleotide for another, the deletion of a nucleotide, the insertion of a nucleotide, or the reordering of nucleotides. 2. Mutations in the DNA can be produced by causes both internal to the organism and from causes in the external environment. These causes are called mutagens. For example, internal mutagens include a certain degree of chemical instability and structural flexibility of nucleotides. 3. DNA segments containing one or several genes are rearranged by certain enzymes, a type of protein. This can lead to the duplication, deletion, inversion, or relocation of a segment of DNA. 4. Horizontal gene transfer occurs when free DNA molecules are taken up by receptor bacterial cells. Footnote 54. This can cause the movement of genetic material across species barriers. Footnote 55. Successful DNA acquisition between species occurs only rarely, but it could, along with DNA rearrangements, be a possible explanation for sudden new properties in evolving organisms. Footnote 56. Genetics has had a great impact on the theory of evolution. Previously, organisms were described by their physical, morphological characteristics, but now their genetic codes can be compared. Genetics, quote, has turned modern biology from a descriptive, morphological, qualitative discipline with no theoretical background into a highly sophisticated, quantitative science based on gigantic, exhaustive, whole genome DNA datasets, end quote. Footnote 57. This impact led to new formulations of the mechanisms of evolution beyond the selectionist theory. While this theory continues to be upheld, now expressed in terms of molecular biology, there are a number of others that have developed. This is due in part to a lack of consensus on the degree to which the above mechanisms of variation contribute to evolutionary change. These other theories include the neutral, nearly neutral, and neo-selectionist theories. Non-Darwinian theories. As molecular genetics began to describe protein and gene sequences, the neutral theory of Mutu Kimura was developed. Consider first that the individuals of a population contain genetic material that during reproduction is recombined to form new organisms with new combinations of genetic material. Looking at the population as a whole, genes will be present in different numbers of organisms from one generation to the next. For example, a gene A may be found in 40% of the first generation and in 45% of the next generation. Because it cannot be predicted which members of the population will reproduce with each other, this change in genetic material of the population, called the gene pool, is said to be due to chance. This mechanism of change is called genetic drift. Footnote 58. The percentage of a single gene will fluctuate from generation to generation until it becomes so prevalent that all members of the population have it, 100%, or it becomes so rare that no member has it, 0%. When either occurs, the gene is said to be fixed or lost, for that gene can no longer fluctuate. It is either in all members or in none. According to the neutral theory, quote, the main cause of evolutionary change at the molecular level, change in the genetic material itself, is random fixation of selectively neutral or nearly neutral mutants. End quote. Footnote 59. For example, a neutral mutation may produce a new form of a gene A called A1 in a single member of a population. Gene form A has a much higher percentage in the population than A1 does. But over generations, genetic drift causes the two to fluctuate in percentage to the whole population, and it may occur that A1 becomes fixed at 100% and the original gene A is lost. A change has occurred in the population due to neutral mutation and genetic drift. Footnote 60. While selectionists stress selected advantageous mutations as being the main mechanism of evolution, the neutral theory holds that advantageous mutations are rare. Instead, most mutations are neutral with regard to natural selection. A similar theory is the nearly neutral theory that takes into account mutations that are, quote, intermediates between neutral and advantageous, as well as between neutral and deleterious classes, end quote. Footnote 61. In these theories, the rate of evolutionary change for the population is equal to the neutral mutation rate. This means that at the molecular level, natural selection of advantageous mutations does not explain evolution. Since the neutral and nearly neutral theories take the emphasis off natural selection, they are sometimes called non-Darwinian. The neutralist/selectionist debate continues to be discussed as to which best explains the evidence. Footnote 62. For Darwinism and neo-Darwinism, natural selection, or survival of the fittest, is seen as the main mechanism controlling evolution. With the neutral and nearly neutral theories, chance neutral mutations and genetic drift are emphasized as the main mechanisms. This led some to understand evolution as a survival of the luckiest. Footnote 63. Neo-selectionist theory. In 2006, a new theory based on the study of the amounts of G and C base pairs was proposed, seeking to integrate the selectionist and neutralist theories. The neo-selectionist theory, quote, reconciles the nearly neutralist view of point mutations with selections at the regional level, end quote. This theory posits a series of steps. First, neutral mutations occur that accumulate together in the DNA. Then a point mutation will add to the previous ones so as to cause changes in the larger region of the DNA structure around it. This is called a critical mutation. It is this final mutation that causes a large enough change to the genetic structure to produce advantageous or deleterious characteristics in the organism that are then selected or not by natural selection. The time during which the neutral mutations are building up and the organisms remain stable is recognized as a conservative mode of evolution, while the appearance of a critical mutation leads to a transitional mode. Head of the research team that produced the theory, Giorgio Bernardi, calls it also an ultra-Darwinian theory, since the neutral and nearly neutral changes are eventually controlled by natural selection. Bernardi observes that, quote, the neo-selectionist theory brings us back from Kimura's survival of the luckiest to Darwin's survival of the fittest, end quote. Chance still plays a role through environmental events, genetic drift, and the neutral and nearly neutral mutations, but it is coupled with the more deterministic role of natural selection. Footnote 64. Gene flow. Thus far, three main mechanisms of evolution have been described: mutations, genetic drift, and natural selection. A fourth mechanism that is present in each of the previous theories is called gene flow. Footnote 65. This is the transfer of genes from one population to another population. Migration of organisms into or out of a population can change which genes are carried to the next generation of the population. Since migration puts separate populations genetically in contact with each other, it tends to lead the two populations to have more similar gene pools, preventing the two populations from developing unique characteristics from each other. Footnote 66. As has been seen from the multiplicity of evolutionary theories, there is no complete agreement on the degree to which certain mechanisms of evolution contribute to the overall system of evolutionary change. This does not mean that the main tenets of evolution are in question. Rather, each of these theories agree on the main pillars of evolution, of which there are three. 1. Variations due to mutations in the DNA drives evolution. Without the variation, there would be no differences to choose. 2. Natural selection directs evolution. The direction is determined by which organisms survive to the next generation. And 3. Isolation modulates evolution. The organisms that can produce offspring are affected by geographic proximity to each other and by the ability to reproduce or not. Footnote 67. As noted before, these mechanisms are ultimately meant to describe how macroevolution occurs. At the same time, they are mechanisms that produce microevolution. In fact, the majority of the research from which these theories of mechanisms are produced occurs from experiments that observe changes within populations rather than changes that produce new species. Footnote 68. Macroevolution and speciation. The previous sections of this chapter have discussed the mechanisms that produce changes in organisms over time. This research observes changes within populations. These observations must then be applied to the question of, quote, change between populations and species, end quote. Footnote 69. In order to understand how these mechanisms are applied to changes between species, one must first consider what a species is. The scientific definition of species is actually debated among scientists. They agree that a species is, quote, the smallest evolutionary independent unit, end quote, meaning that the mechanisms of evolution, mutation, genetic drift, natural selection, gene flow, act on that group of organisms separately. The flow of genetic material is contained in this group of organisms. To fully define species, one must have a criterion for determining an independence of gene flow. Footnote 70. There are 3 definitions of species that use different criterion for determining independent gene flow. The traditional definition of a species, called the biological species concept, uses the criterion of reproductive isolation. That is, organisms are of the same species if they can reproduce fertile offspring. If they cannot reproduce fertile offspring, they are of different species, thus preventing gene flow. Unfortunately, this definition has several limitations. First, it only applies to organisms that reproduce sexually. Second, if populations are isolated geographically, and it is not possible to artificially test if their offspring are fertile, one has to make a subjective judgment as to whether they are isolated reproductively. Third, it cannot be tested with fossilized organisms. Fourth, it is difficult to apply in many plant groups that have hybridization occurring. Because of these limitations, 2 other definitions of species have been proposed. Footnote 71. The phylogenetic species concept is based on the criterion of monophyly. Under this criterion, quote, species are named on the basis of statistically significant differences in the traits used to estimate the phylogeny, end quote. A phylogeny describes the evolutionary relationships among organisms. This family tree is determined by chosen traits. Footnote 72. The divergence seen in these traits, genetic and/or morphologic, between the 2 species can only have occurred due to isolation and gene flow. This criterion has the advantage of being able to be applied to any type of organism. One disadvantage is that traditional species names may need to be changed, and many more species will be differentiated. The final proposal for the definition of species is the morphospecies concept. This definition uses the criterion of morphological differences alone. The advantage of this definition is its wide application. It is often used in paleontology where, quote, reproductive isolation and well-estimated phylogenies are lacking, end quote. The disadvantage is that, if it is not applied carefully, it can lead to incomparable differences between researchers. Also, it limits the type of traits that can be used to distinguish species. Color, soft tissue, habitat use, or genetics, for example, cannot be used. Footnote 73. As often as possible, more than one definition will be applied, but in many cases, only one is possible. At times, the different definitions will lead to different results, whether in distinguishing species or in creating phylogenies. The process of speciation. Having looked at what a species is, it remains to understand how new species come into being. The process by which new species are produced is called speciation. Speciation forms the bridge between microevolution and macroevolution. Freeman and Heron write, quote, "Classically, speciation has been hypothesized to be a three-stage process. An initial step that isolates populations, causing genetic isolation. A second step that results in divergence in traits such as mating system or habitat use. And a final step that produces reproductive isolation. Recent research has shown that the isolation and divergence steps that initiate speciation frequently take place at the same time and in the same place." End quote. Footnote 74. This gives three steps to the speciation process: population isolation, divergence in traits, and the production of reproductive isolation. In the first step, according to what is called the allopatric model, a small population on the edge of a species range becomes isolated from the rest. This can occur due to dispersal. A species becomes dispersed over a wide area with individual members isolated from the original population founding new colonies. Vicariance. An event occurs that splits a species distribution into isolated ranges. Or polyploidy. Changes occur in the chromosome number, making reproduction incompatible. The first two causes are environmental, while the third is genetic. The third, polyploidy, rarely occurs in animals. Footnote 75. Population isolation creates the condition necessary for divergence to occur between the original species and the small population separated from it. Divergence can occur when genetic drift, natural selection, and sexual selection act on mutations in the same populations. Since the new population is isolated from its original, the two are completely independent in how the mechanisms of change occur. While it is hypothesized that genetic drift should produce rapid genetic divergence in small isolated populations, the role of genetic drift in speciation is controversial. It has been discovered that, quote, "only very rare alleles, forms of genes, tend to be lost due to drift." End quote. For great changes to occur, populations have to be very small for a significant time. Even so, populations that seem to fit this category have shown no signs of speciation. Sexual selection is hypothesized to occur when a population changes the way it chooses or acquires mates, leading to rapid differentiation. Evidence for sexual selection is not yet conclusive. Instead, quote, "natural selection is becoming recognized as the most important force promoting the divergence of populations." End quote. Footnote 76. The final step, reproductive isolation, is reached when the divergence is great enough between the original species and the isolated population that even if they come back in contact, gene flow cannot occur between them. A new species is thereby developed. If the isolated population comes back in contact with the original population before great divergence has occurred, interbreeding of the two diverged populations can produce fertile offspring. It is proposed that while the two populations have characteristics best suited for the areas in which they diverged, hybrids of the two would tend to produce offspring less adapted to either area and thus not as likely to be selected naturally. Natural selection would then tend to select changes in both populations that prevent hybridization, since hybrids would be less likely to survive. This process of reinforcement would ensure that reproduction occurs within each population separately. What if the hybrid actually is better fitted to survive and reproduce? It is contended that the hybrid could develop into a new species. Quote, "reinforcement can and does occur, but it is not essential for speciation." End quote. Footnote 77. Freeman and Heron give a number of concrete examples of researchers studying this process of speciation. These studies document a divergence of separate species using the second definition of species. Even though these species may have a very low gene flow, which would define them as a single species under the traditional definition, they are defined to be separate species under the phylogenetic species concept. Origins and transitions. Using molecular biology and morphological characteristics, phylogenies are constructed, hypothesizing how life began and how it progressed to more and more complex forms. Theoretical chemist Peter Schuster notes, quote, "A persuasive finding, if not the most convincing evidence of all, for the correctness of the theory of evolution is the fact that the phylogenetic tree based on morphology and the molecular genetic family tree of life forms, with some exceptions, agree down to the most minute details. Since the two methods of reconstructing the history of life are independent of one another, they form the two legs on which current evolutionary biology stands." End quote. Footnote 78. This construction of the history of life begins with some primordial form, which may have been catalytic RNA molecules, all the way to the diverse species of organisms living today, including humans. Footnote 79. In order to reach the level of complexity of human beings, scientists recognize 8 major transitions that would have to be made. See Appendix C. Of these, the transition from cells without a nucleus to cells with a nucleus is the most plausibly explained. Footnote 80. It is unknown exactly how all these transitions occurred. The next section will consider the final transition from non-humans to humans. Theories of human evolution. Models of human evolution begin with the underlying model of evolution that all species of organisms are derived from species that have come before them. Theories of human evolution began first with the comparison of physical traits between humans and the higher primates. This was later coupled with findings using the techniques of molecular biology, which discovered high similarity between the DNA sequences of humans and chimpanzees. While chimpanzees have one more chromosome than humans, when one compares chromosomes, about 95% of the base pairs are identical. Footnote 81. As noted previously, this similarity, both morphological and genetic, leads the evolutionary scientists to propose a common ancestor. Further, a number of fossils have been found which, based on morphological characteristics, are placed in a category of beings leading up to humans called prehumans. These pre-human fossils are categorized into 7 genera and 14 species. It is not known exactly which of these marks the last common ancestor of humans and chimpanzees, but it is hypothesized to be perhaps one of 3 candidates. Footnote 82. Recreating the history of human evolution as best they can with the limited evidence, scientists propose the appearance of the genus Homo in the Omo River area in Ethiopia, Africa. There were 2 species of Homo, Homo habilis and Homo rudolfensis. From these developed the species Homo erectus, which in turn developed into 3 species isolated geographically. One of these 3 was Homo sapiens, which is the species classification for humans today. Footnote 83. This proposed phylogeny is not without its questions. French anthropologist Yves Coppens admits that, quote, "the phyla that we are trying to build and draw with our bones and teeth is only based on anatomy and morphology, and it is a bit light." End quote. Footnote 84. It has not been possible to utilize the findings of molecular biology to form the relationships among the fossil remains and living humans, since DNA cannot be extracted from the fossils. Instead, one must rely on physical traits alone. This leads to several questions in Coppens' mind, especially surrounding the transition from Homo erectus to Homo sapiens. Quote, "As far as Homo sapiens are concerned, I am not happy. About the origin of Homo sapiens, it is a bit confusing." End quote. His question is not, did man evolve, but exactly how? There are enough fossils to tell that, quote, "there is really a transition between Homo erectus, who is completely Homo erectus, and Homo erectus, who is a bit sapiens, and Homo erectus, who is so sapiens that he is sort of sapiens with some erectus in it." End quote. Footnote 85. Where exactly do humans appear in this phylogeny? There seems to be disagreement on this point as well. Coppens understands the appearance of the genus Homo to be the appearance of man. Quote, "Homo looks like a product of nature, a necessary adaptation to a climatic change. but is a human being with, for the first time, this capacity of knowing that he knows. He is a human since this beginning. So it is sort of a discontinuity in a continuum. End quote. Footnote 86. Freeman and Heron also concur that 5 fossil specimens labeled with the genus Homo are, quote, uncontroversially human, end quote. Footnote 87. Anthropologist Fiorenzo Facchini disagrees with this placement on the basis that, quote, a skeletal remain attributed to the genus Homo because of its anatomic features does not necessarily imply that it represents man in a philosophical sense, i.e., a thinking man. End quote. Footnote 88. Facchini argues that the appearance of man should not be based on morphology alone, but on external signs that indicate the individual has man's higher capabilities. These external signs include evidence of human technology distinguished by, quote, the complexity of actions by means of which this technology is performed, and the capacity of improving and innovating technique, and the significance assumed by the products in the context of everyday life. End quote. Footnote 89. Professor of Cognitive Neuroscience Stanislas Dehaene seems to attribute the appearance of humans with that of the species Homo sapiens. Footnote 90. There are also questions as to how many different species the Homo specimens actually represent. Footnote 91. Summary. This chapter has by no means been an exhaustive look at the research that has gone into evolutionary studies. The evolutionary theories of natural selection theory, neo-Darwinism, neutral, nearly neutral, and neo-selectionist theories attempt to explain how descent with modification may occur, centering around 4 main mechanisms: natural selection, mutation, gene flow, and genetic drift. While these may be the main evolutionary theories, both historically and now, other proposed theories also exist. While these other theories often agree that descent with modification has occurred, questions are raised concerning the proposed mechanisms. Do these 4 mechanisms, which seem to adequately describe microevolution, sufficiently explain macroevolution? There are legitimate questions about the limitations of the current evolutionary theories. For example, the lack of explanation of the major transitions and the very limited observation of speciation. Also, can these mechanisms alone explain events such as the Cambridge explosion, a time of rapid increase in the diversity of existing organisms, or the development of certain molecular systems containing complex interacting parts? Footnote 92. Is it statistically probable or even possible for the higher organisms that are observed to be produced based on the current mechanisms? Footnote 93. Insofar as these questions remain within the methods of science, they need to be discussed by scientists. This is why this work refrains from critiquing scientific theories as to their truth. The truth of a theory of evolution, insofar as it is a science, is a scientific question that remains outside the scope of this paper. This chapter has given an overview, including the initial observational evidence, formulations of theories to explain the evidence, the role of molecular biology in explaining variation, particularly within populations, the application of evidence within populations to the formulation of new species, and the theories of human evolution. It is necessary to know such background in order to clearly understand its interaction with philosophy and Catholic theology. End of Chapter 2 of Evolutionary Science and the Catholic Church by Reverend Mr. Kevin W. Vogel, read by Father Kevin W. Vogel.